Welcome to the Metabolic Link, a medical and science-focused podcast that explores the common thread of metabolism in health and disease. This is where science meets society. Hi, and welcome to another episode of the Metabolic Link. We're excited to be back for another research dive, or as some people are calling it, journal club out there. Really exciting. I'm Victoria Field, one of your co-hosts, along with my fellow co-host, Dr. Dominic D'Agostino, Dr. Angela Poff. Before we kick off, why don't you guys give super brief little background, who you are, what you do. I'll go, and then we'll dive into the fun stuff in these papers that we've got today.
My name is Angela Poff. I am a research associate at University of South Florida. I work alongside Dom there in his lab dedicated to metabolic medicine. So we study all kinds of things related to how metabolism plays a fundamental role in the development of disease and how we might be able to target it therapeutically for the development of novel treatments. And a big interest of mine is cancer metabolism in particular.
Dom D'Agostino, background in nutrition, and then I was formally trained in neuroscience and also in physiology, studying how the brain controls autonomic regulation, and have a big interest in moving the science that we've done for many years into human application and also education outreach. So then hence this podcast. And I'm so glad that you are joining us, and we have a lot of things to share today on those topics.
Awesome. And I'm Victoria, extensive background in fitness and nutrition, and also implementation of ketogenic diets, specifically with cancer patients. Also co-founded a Metabolic Health Initiative with Angela, and we co-organized Metabolic Health Summit, of course, with Dr. D'Agostino over there. And excited to dive into the discussion today because we've got a couple of really, really interesting papers. One touches on Alzheimer's, the other on MS. The first, though, Angela's going to be kicking this off for us and kind of take us through the research, and then we're going to have a little group discussion around it. But the paper is called Warburg-like metabolic transformation underlies neuronal degeneration in sporadic Alzheimer's disease. And so, Angela, do you want to kick off this paper, and we'll then explore it further?
Sure, happy to. Okay, so this is a really cool paper. It was published in September of 2022, so a few months ago, published in Cell Metabolism. I was blurring out my paper here, but and it comes from a collaborative group of individuals, researchers from some different universities, including the Salk Institute, University of Colorado, UC San Diego, and essentially, the premise of this study was to try to understand some of the molecular mechanisms that are underlying how a healthy neuron ends up being converted into an unhealthy neuron in sporadic Alzheimer's disease. So we hear a lot about the genes where there are known mutations or just the possession of a certain allele, a certain version of that type of gene is linked to developing Alzheimer's disease. So those genes include things like amyloid precursor protein, APP, presenilin 1 and 2, ApoE4. And these genetic factors are inheritable. And so the cases that are caused or related to these specific genetic conditions are called familial Alzheimer's disease, and it's because they're inherited. And so, but the vast majority of Alzheimer's cases, like over 95% of them, are actually considered sporadic. And these are the more classic late onset kind of typical presentation of Alzheimer's disease. There's a lot about the way that sporadic Alzheimer's disease develops that we don't understand. But we do know there are many significant metabolic alterations that are found in individuals who have sporadic Alzheimer's disease. So if we look at their CSF, cerebrospinal fluid, you can see modulation of a lot of the metabolites there. And if you look at the expression of metabolites in the brain tissue from a patient post-mortem that has died from Alzheimer's disease, you see these changes as well. But currently, a lot of this is really just like correlative. We can understand that these are biomarkers, but we don't know exactly if they're playing a role in the progression or the development of sporadic Alzheimer's. So in an attempt to kind of untangle some of these mechanisms, these researchers generated what are called induced neurons from some fibroblasts from patients using expression of specific transcription factors. So to explain that a little bit, induced neurons, they're sometimes called induced neuronal cells because they're not truly neurons, but they function, behave very similar to neurons. They can be generated in the lab from using other types of cells like fibroblasts. These are connective tissue progenitor cells in the skin. And you can do this by causing the expression of certain genes, and in particular transcription factors, which are proteins that cause the DNA to express specific genes. And they will activate this kind of like gene expression pattern that is associated with a differentiated or mature cell type. So in this case, you take fibroblasts, you express certain transcription factors, and now all of a sudden that cell has kind of converted into a neuron. So those are called the induced neurons. And so these fibroblasts are basically being reprogrammed into neurons or neuron-like cells that you can then study. And so it's been shown that Alzheimer patients, if you create induced neurons from Alzheimer patients, these induced neurons will lose their mature neuronal kind of markers, and they regress to this more immature kind of state. And the process of this happening is actually strikingly similar to the way that cells transform into cancer cells during oncogenesis or cancer development. So we term that transformation from a healthy cell into a cancer cell, we call it malignant transformation. And just like you see there, these cells begin to kind of get rid of their mature or differentiated characteristics. So these specific little markers, they like to hold up and say, hey, I'm a mature neuron. And so they start getting rid of those things and they regain a more primitive state. And so the goal of this was to understand what are these mechanisms that are causing this neuronal hypomaturity in these cells, because it could represent a really early step in Alzheimer's development that could potentially be reversible or targetable. And the overall kind of result was that they found PKM2, this specific metabolic enzyme and nuclear factor that is very common in cancer, is also expressed in Alzheimer-induced neurons, and it basically is responsible for causing this loss of their resilience that occurs. So to kind of get into the meat and potatoes of exactly what they did, so they took punch biopsies from Alzheimer's patients, control donors, and they retrieved dermal fibroblasts from them, made them into a cell culture line, so grow them up in petri dishes so they could study them. And they had controlled for age, sex, and ApoE4 status between the patients and the control donors to make sure that you didn't have these kind of confounding variables. They overexpressed transcription factors ACL1 and neurogenin-2 to generate these induced cortical neurons. And that worked perfectly. After about 21 days, they're behaving more and acting like neurons. And then they also took neurons from post-mortem brain samples from patients who had died from Alzheimer's disease. And essentially, they performed something called weighted gene correlation network analysis, where they looked at the genetic expression of all of these different cells, and they're trying to determine, okay, how are the gene expression patterns different, or how are they the same between these cells? And they found that there were a lot of distinct genetic signatures that had overlapping changes between the Alzheimer's induced neurons and the Alzheimer's postmortem samples. And so this suggests that these pathways they identified could be really important in that transformation of a healthy neuron to an Alzheimer's neuron. And one of the major ones they found was pyruvate kinase. So This is an important enzyme in glycolysis, catalyzes the last step of that pathway. And there's an isoenzyme of pyruvate kinase called PKM2, which is essentially just a different enzyme, but it catalyzes the same reaction. That is not typically expressed in mature differentiated cells, but it is often in cancer. It's overexpressed and turned on in most tumors. And it drives this completely different metabolic phenotype within these cells to make it Warburg-like. And so that was the major distinguishing characteristic, was that alteration in this pathway created this Warburg-like phenotype in these cells.
Maybe go back to the basics for those who don't really know what Warburg-like is exactly.
Yeah.
And I know you guys are very familiar, of course, on the cancer side of things and what that looks like. So for those who are watching or listening, it'd be great to get a good breakdown of that.
Yeah, and I have a quick couple comments. This is the first time I've heard about the Warburg effect. When you sent me the paper, gave me my reading. The opening Warburg-like metabolic transformation, I'm thinking it's going to lead me to cancer, but Alzheimer's disease. So this is kind of like a new idea that's very interesting. And also the amount of expertise that went into this publication is amazing. Like if you look at all the authors on this, so it really adds to the credibility of the study and the findings. There's one particular author, Fred Gage, and I remember we had a class on stem cells, and this ties into some of the figures we'll be talking about. In the 1990s, like 1998, 99, they were taking bone marrow stromal cells, and then not fibroblasts yet, but maybe different types of cells, and transforming them into neurons. And the big thing was that they would express neural markers, but these transformed cells would not be firing action potentials. Mm-hmm. So in this figure, you can see that they can literally take fibroblasts, transform them, mm-hmm. and they become functional neurons, which is really interesting. But yeah, I digress. But I want to let the listeners know that this is a super powerful combination of minds that were on this, and it's a pretty complicated paper. But I think you're doing a great job of breaking it down. Even though, what you're talking about sounds incredibly complicated, but you are breaking it down in a way that's understandable. Yeah.
Oh, thank you, Dom. I appreciate that.
Educated layperson. Yeah.
Yeah. So, I totally agree. It's a really incredible paper. It's huge. I printed it out and it used up all the paper in my printer. Yeah. But they just did so much work to really not just show correlative studies, or the correlation between things that are changing, but to really show that there were functional kind of consequences. And that goes back to the heart of the question that Victoria asked about the Warburg effect and kind of explaining what does that mean. So the Warburg effect is a phenomenon that is characteristic of most cancer cells, wherein tumors will change their metabolic phenotype so that they are consuming large amounts of glucose and not fully metabolizing that through the mitochondria. They are instead shunting it through towards lactate production. And so this is abnormal. Most mature, healthy cells will preferentially utilize the mitochondria for their metabolism. And in most cancers, you see this heightened glycolytic phenotype where lots of glucose is being sucked up and being shunted and specifically towards lactate production. And so that was something they actually showed. So, they identified through these gene signature kind of analyses that PKM2, and glycolysis was upregulated, but then they really, also performed a lot of detailed and really elegant molecular work to demonstrate that the changes they were seeing actually had functional consequences, in this way they anticipated that they would. So really confirming that this global Warburg-like metabolic switch was actually happening. So they saw in these induced neurons, the Alzheimer-induced neurons, that they were exhibiting this huge switch towards glycolysis. They had heightened lactate production. They increased in the amount of several of these glycolytic intermediates. And in fact, glucose uptake itself was extra high in these neurons as well. And so this was really just confirming that you had this Warburg type effect that was being observed in the cells. And it's interesting because, of course, you can kind of, speculate why would that potentially be happening in Alzheimer neurons. And in the cancer field, we talk a lot about like, okay, is this an advantageous adaptation? I think I just merged those words. Adaptation. Yeah, right. Or is it, the consequence of some kind of like damage? Like, is it because the mitochondria are damaged, they're doing this, or are they choosing to do this because it benefits them in some way? And, that's also something, of course, they're kind of questioning here. The authors actually did a lot of really cool work to show that that oxidative phosphorylation was normal. So mitochondrial metabolism was normal in these cells. So they were really thinking that this was, for whatever reason, a phenotype that glycolysis was heightened over and above normal levels of oxidative phosphorylation as well.
Super interesting.
Yeah.
And I think what's most interesting is because what you have on the cancer side, or at least in sort of early stages drug development, and what they're focusing on inhibiting on the cancer front is, PKM2. So if you had something similar on the Alzheimer's side, I mean, what does this mean, right, in terms of the treatment and pathology of Alzheimer's in general?
What is inducing it? It's a very interesting phenomenon. And maybe, with cancer, it's a dysregulation and kind of stress in a disturbance in the microenvironment, and the mitochondria being a major player in the cascade of events that cause the oncogenic transformation to damage respiration with compensatory fermentation, which kind of describes the Warburg in cancer. Mm-hmm. But in this phenotype is not. And I didn't delve into it as much to see the clarity, but you're saying that oxidative phosphorylation continues to go unrestrained or unimpeded.
Yeah.
And then there's an acceleration of glycolysis. And we know that the etiology of Alzheimer's could be a number of different things, but there could be there could be a decrease in blood flow, and these things could potentially create an energetic crisis where the cell is trying to get more glucose or utilize, more glucose. So that's where I'm thinking, but I'm waiting for you to take me down.
Totally. Yeah, okay. No, I completely agree. And that's what makes metabolism studies so complicated. It's because they do, of course, all feed into each other, and then like the metabolic pathways do, and it's all influenced by both, a local, like within the immediate microenvironment of the cell, but then also the tissue, and then systemic changes. And also, in response to signals coming in from the outside, what food you're eating, how you're, exercising, moving, using your nutrients. But yeah, so they actually showed that that most of the indicators of mitochondrial function were normal. So the activity and flux of electron transport chain complexes were intact. TCA cycle metabolite levels were normal. The ATP to ADP ratio was normal. Mitochondrial membrane potential was normal. So all of these things indicating that it was some kind of, flip. What's really interesting, and what they ended up taking it back to your question, Victoria, about like, what does this mean when you think about, could this potentially lead to something that could be therapeutic for Alzheimer's, was they basically, found that they did a lot of more experiments. There was so many experiments that went into this paper. It was really cool. But they performed a lot of experiments that showed that this metabolic switch towards glucose metabolism was driving this immature apoptosis within the neurons. So in Alzheimer's disease, especially towards the end, you have a really catastrophic loss of neurons. So a large amount of neuronal cell death. this is abnormal for neurons. So mature neurons are actually pretty resilient compared to other cells in our body against apoptosis. And it's because mature neurons don't typically, divide and replenish themselves, and they have to make it basically from early development. They have to survive, 80 plus years. So they kind of have this like protection. For whatever reason, when PKM2 was activated, this Warburg phenotype was established. These more immature neurons that were then produced because of that, they had lost this resilience against apoptosis. So, and they were actually able to rescue that with a chemical inhibitor of PKM2. So this is a classic kind of, way that you would try to develop a targetable drug towards something. You show a deficit when this marker is present. If you can inhibit that marker and stop that deficit from appearing, then you've really proven this is in fact causing this. It's actually a causative kind of study versus correlative, right? And that's the beauty of being able to do this kind of work in animal models and cell models. You just can't often establish causation with humans trials. That's why we kind of need all of this. But essentially, they're saying they were able to give a chemical that inhibits PKM2, and they basically reversed a lot of those Alzheimer-like features in those induced neurons. So they weren't exhibiting this big glycolytic phenotype anymore. And perhaps more importantly, they weren't exhibiting heightened apoptosis or cell death anymore. And if you think about that substantial loss of neurons really contributing to the overt pathology of a patient with Alzheimer's towards the end, that could be a really promising target to, could we prevent this early on by not allowing these cells to activate PKM2? And that's what I love about metabolism is it's opens up a path for so many different ways. We talk a lot about lifestyle changes that can impact metabolism, but metabolic pathways are also completely targetable by pharmaceuticals and things like that. And I think it's really important that we're exploring all of those avenues. So it was cool to see that this could be a really viable path towards developing some kind of effective therapeutic for the development of sporadic Alzheimer's.
And can you imagine if you combine that possible drug treatment with all the lifestyle changes?
Yeah, exactly.
Sleep, the reduction of chronic inflammation, all the things that, researchers like Dr. Dale Bredesen, he provides a variety of contributors beyond just sort of targeting nutrition, including sleep, including environmental toxins, including pathogens and dealing with those things. So, it's really exciting to think about if you were to have something like an effective pharmaceutical treatment like that, we're also able to implement some of these other things day to day. The future's bright, right? So hopefully this is the step in the right direction for Alzheimer's.
Yeah.
So the Warburg transformation from PKM1 to PKM2 would lead to proportionally more lactate, right? Yeah. Could we think about this as a way that the brain is trying to generate more lactate as an alternative fuel? From
Possibly.
That perspective, right? That's the first thing I thought about with PKM2 is just, generating lactate, and then cancer cells throw it out into the microenvironment and then acidifies it, and then that drives invasiveness and metastasis. But could the energetic crisis or whatever is triggering this, and that's a whole nother discussion, I guess, be leading the bioenergetics of the neurons to use an alternative fuel? In this case, it would be lactate, right?
Yeah.
So, that's, I kind of think about the whys, and if we know the whys, then we can go upstream in figuring out, the paths.
Yeah, I think that's totally possible. It's fascinating when you think about how common it is that you have this kind of altered metabolism, especially like in neurological diseases and neurodegenerative diseases. And you have this kind of, on like a systemic level, you see like reduced glucose metabolism. So this is really interesting because, for example, Dr. Stephen Cunane has done a lot of work showing that in patients with mild cognitive impairment and Alzheimer's disease, you see reduced glucose metabolism. or the reduced ability to metabolize glucose, but ketone metabolism is normal. So why would, this is talking about a really early change where you're activating this glycolytic phenotype. Could it be like it is an early compensatory switch? So whatever is, there could be something initially that's trying to, you have this hypometabolism, this inability of the brain to have enough energy. And so perhaps like in cancer, which some people would argue, you activate PKM2 and just, put your foot on the gas pedal in terms of trying to increase flux and through this energetic pathway. But eventually maybe it's not enough. And so that might explain why on the molecular level, you're seeing hyperactivation of this glycolytic pathway, but on a global level, you see reduced glucose metabolism. And I thought it was cool though, to see that in this study as well, oxidative metabolism and mitochondrial metabolism was intact because ketones require mitochondria to be metabolized. And Dr. Cunane has shown that ketone metabolism is intact in these individuals. And so seeing that on a molecular level, in these neurons, you're also seeing normal metabolism supports this idea that like, wow, switching to a metabolic substrate that like ketones really could be impactful because at least, it seems that part is still functioning even in these neurons.
So I go back to thinking about, and I haven't really dug into this paper like you did, but so glucose metabolism in the brain is impaired in TBI, and pretty much every neurodegenerative disease is pathophysiologically linked to impaired glucose metabolism. And that could be from pyruvate dehydrogenase complex, PDH, but also there's an internalization of the GLUT3 transporter, and which could then trigger an energetic crisis for the potential transformation of a PMK1 to PMK2, and that transformation would potentially be a result of an energetic crisis that's signaling to ramp up glycolysis, and that transformation could be kind of a path to maintain glycolytic activity and glucose metabolism, but also a way to make proportionally more lactate. And it looks like, I'm not sure they quantified it, but you're producing at least about double the amount of lactate from some of the graphs and things. So you have preservation of oxidative phosphorylation, which would make ketones an available, readily usable source. And then perhaps maybe a glucose hypometabolism is a hallmark characteristic of Alzheimer's disease. So, so there's an energetic crisis, and then it's ramping up through the microenvironment, a transformation of PKM. So, and then that is proportionally making more lactate, which then functions also as an alternative fuel. It'd be interesting so I'm always, I don't know if Angela, you probably remember me talking about this, but lactate was one of my original pet projects that I had back in my postdoc, and alpha L polylactate, kind of because it was in the supplement I was using called Cytomax. And I think there's some weird patent there, but it's amazing. Like, I use it when I was mountain biking. I don't know whatever happened to that, but it's alpha L polylactate, and it delivers lactate to the muscles, but also the brain. So it just it kind of reinvigorates my interest in alternative fuels for the brain, so ketones and lactate. And I'm a little bit biased, but I think this is a metabolic transformation strategy to increase lactate in the brain.
I think it totally could be. I would, I wouldn't be surprised. That's really cool. It makes me kind of think of, you could see parallels between like what happens in insulin resistance, right? That like there's this compensatory increase in insulin to try to get more glucose into the cells because it's just the receptors are the cells think that they don't have enough glucose, but it's not because they actually don't, right? And then in this case, maybe there's some kind of hypometabolism that is causing the cells to need to upregulate, but in the end, it's not enough, right? And you have this kind of like burnout and eventual, death of the neurons, or in like extreme cases of type 2 diabetes, potentially loss of pancreatic beta cells. So it's cool to see how far the body can go to try to keep things functioning. And a lot of this is through using the metabolic pathways to, to support until it can't any longer.
Yeah, it's encouraging because there's not too many options on the Alzheimer's front or MCI. I mean, you look at some of the drugs available right now, and so shifting gears and kind of focus towards the more metabolic approach, that maybe is also inclusive of lifestyle or a combination of, pharmaceutical therapeutics or what have you, I think is really encouraging because we don't really have many things, especially something that maybe might help early on in preventing this from further progression. So
and
really,
that makes me think also of, we talk a lot about how Alzheimer drugs have largely failed. Been a lot of money, a lot of effort towards trying to develop pharmaceuticals for this. And but that has largely focused on trying to address like the amyloid plaques and the tau tangles. So this is a totally different strategy. And, hopefully it could be more promising, especially come on the heels of how long we've not had big success with development of pharmaceuticals for Alzheimer's disease.
Yeah. So the Shikonin, I don't know if so that's the activator, and I was unfamiliar with it, but it's kind of a dye. It comes from Barrage. It's used in the cosmetics, red, brown, food additive from regions of Australia, and the R enantiomer of L-conin is known as Shikonin, and the racemic mixture of the two is Shikalanin or Shikakalin. I'm not pronouncing.
Maybe explain for people who might not know exactly what you're diving into there, Dom.
So naturally, I look up that they use this Shikonin to increase the enzymatic activity, and of course, people are selling it as food supplement. And I think it's even on Amazon, maybe. Yeah. So, so it just kind of sending me down that path to think about, various ways to augment metabolic control within the brain. This particular enzyme is very interesting. So,
so that was the PKM2 inhibitor that they.
Oh, PKM2 inhibitor. Yes.
Yeah. Which would therefore activate normal, pyruvate kinase activity. So,
So reverse the transformation.
Yeah.
So one could think like in the Warburg effect with cancer, I think the oncogenic transformation leads to a de-differentiation of the cell type in cancer that could be a liver cell, a kidney cell, a muscle, which is a little bit more rare, but a skin cell. So there is a similar de-differentiation kind of, but it's de-differentiating it into a cell that has some unique metabolic characteristics, but also isn't that cell farther on the paper, and apologies, I didn't dig into it that much, but doesn't it make the cell more vulnerable to an apoptotic or apoptosis, and also makes the cell vulnerable, selectively vulnerable in a way for death, right?
Which is super interesting because when you have this transformation that occurs in cancer cells, it's associated with a robust resilience to apoptosis. So that's strange because, with a cancer cell, this you want apoptosis to happen in these cells. You want apoptosis is cell death, programmed cell death. Cancer cells are really good at avoiding signals, like anything, the house can be on fire and they're like, nope, everything's just fine. They're like that dog in the bar with everything's on fire. They are. And so, they avoid these apoptosis signals when they shouldn't. And then in this case, in Alzheimer's disease, you have this transformation that's also associated with the same kind of metabolic switch, but you now have cells that are more sensitive to apoptosis and more likely to undergo apoptosis. And it reminds me too of, just trying to find ways to support neuronal survival there's research from preclinical studies showing that ketone metabolism will increase the resistance of neurons towards activation of the mitochondrial transition pore, which is what forms and initiates apoptosis. So ketones in the presence of neurons being treated with some chemical that would induce apoptosis are more resilient to apoptosis when ketones are present, because it specifically inhibits a key step in apoptosis. So like another kind of mechanism that would support a metabolic targeted kind of therapy where this, ketones could potentially offset some of this increased susceptibility to neuronal cell death, which is pretty cool to think about.
Really interesting.
Yeah.
And it kind of just makes me think of some cancer research just showing how implementation of a ketogenic diet or ketones might improve the efficacy of some of the cancer treatments that some people are going through. So I don't know, this thought of combination therapy for something like Alzheimer's, tackling it with lifestyle and a targeted drug like what they're looking into here is really exciting. I'd say.
Totally. I agree.
Awesome. Well, if you want to check out this paper and take a deep dive into the weeds, you certainly can. We're going to be linking this paper in the description of this podcast. But why don't we shift gears into the MS study that we've got on the table here. And this study is titled Phase 2 Study of Ketogenic Diets in Relapsing Multiple Sclerosis, Safety, Tolerability, and Potential Clinical Benefits. A really exciting paper here. Dom, do you want to kick this one off for us?
Yeah, absolutely. So I do teach neuroscience in the medical neuroscience class, and we do cover a little bit about MS and the etiology of MS. And it's kind of like not much to teach there, right? Because we don't know the etiology of MS. The best we can kind of know is that, we can go very deep into the weeds about the demyelination effects, and what that causes, the effects of unmyelinated cell in regards to the cell membrane and the how it leads to. I teach about the capacitance and the membrane potential and how that becomes disrupted and decreases the signal. But the etiology of MS is largely unknown, and there's autoimmune hypothesis and viral hypothesis, but it's interesting that younger people are getting MS. And when I started, when you sent me the paper to read and to cover, I started digging into MS a little bit more, which I do from year to year to update my slides. But what is really striking is that younger and younger people are getting MS. Like kids, there's pediatric MS. So that's paper came up on pediatric MS, and that kids are younger, but it tends to be associated with metabolic dysregulation and obesity and the kids that get MS. And so it's really interesting. Yes. So about 2/3 or more are women. And so MS, taking a step back, is a demyelinating disease that happens. You get white matter hyper intensities throughout the brain and throughout the spinal cord too, and these show up as degeneration, a demyelination, and so this paper is like you said, a phase 2 study. So a phase 2 study. So I got really excited about a phase 2 study because that's really focused on efficacy. But this is like a phase 1.5 study. It's an amazing study, and there's many aspects to it that are very, very remarkable and unlike any other ketogenic diet study that we have covered. And I think what's really remarkable is that the dietitians, Deanna Gulati, I think she's part of Denise's Potter team. She was part of the creating the ketogenic diet and administering what was a modified Atkins diet. They had like 83% of the people follow the diet for MS population. That's amazing. But a phase 1 study is more about safety, and a phase 2 study is really phase 2 study is about 100 to 300 people, and it's like, you know, safety, but also efficacy and tolerability and things like that. And then this study does show some clinical benefits. And then just for the listener, phase 3 is like 1,000 to 3,000 people, right? And then a phase 3 study after that, then it goes to an FDA review. And now there's like phase 4 studies where it's like, you know, they're doing it with the vaccines where you have tens of thousands of people, right? So they did a phase 1 with a modified Atkins and showed some feasibility. And this is a phase 2 study. And another step back, this journal is a very good journal, a journal of neurology, neurosurgery, and psychiatry. It's like an impact factor of almost 14. So very good journal using a modified version of the ketogenic diet. And it was kind of tailored to the patient. And I think that had a lot to do with the compliance and adherence. And so then again, so MS is, the etiology is kind of unknown, but it does seem to be, this paper is really suggesting, we can say right off the top, MS is responsive to ketogenic therapies in a number of different ways. So, and we can kind of delve into that a little bit more. MS occurs, so there is a pediatric, but classically MS occurs from the age of about 20 to about 50. I think maybe a mean age of about like 40, like late 30s or 40s or something like that. So, so in this case, they use a modified ketogenic diet. There was 65 subjects with relapsing MS, and it was a 6-month study that they did. And they looked at to ensure that they were adhering to the ketogenic diet. They did not do blood ketones, which I can dig them on. But, you know, if you talk to the neurologists in the field of epilepsy, they'll tell you that urine ketones are a pretty good indicator of if you're in a state of ketosis, you know, and everybody pees every day, hopefully. So you don't, it's extremely non-invasive. So you have, you know, urine that you can measure every day. And also like the urine strips that you could use, they use the Bare keto sticks. So which just measure urine acetoacetate. But you could use like the multi-stick SG, which measures like, you know, a dozen or 20 different things. So, I mean,
is this for like just initially, Dom, in testing urine, or are they recommending ongoing in urine? Because typically ketones kind of start to wane in urine after a while, right?
Well, you know, in people that are kids or people that are really following a strict calculated ketogenic diet, the ketones do go up and then they come down, but they're always there in someone that's truly following like a ketogenic diet, like at least in kids. But what the dietitians did, and this is part of the dietitian that again was part of Denise Potter's team, they had them measure urine ketones every single day, and then they date it, and then they take a picture of it like every single day. So you have daily measurements telling someone, I mean, this study really gave me a lot of ideas that I wrote down, but telling someone to measure ketones every day. And then I think they had to have like 85% of the time or something like that had to be in a positive state of ketosis. And otherwise they're not adherent to the ketogenic diet. So I thought that was a really cool, at first I was like, urine ketones, come on. I mean, there's so many different technologies, but being able to get, I mean, they could do like blood ketones once a week or something, but you know, the patients were at home and they had the urine and they could test. But so they had, you know, 65 subjects they ended up with, and they had really positive effects on fatigue, on depression. So we talk about metabolic psychiatry. I mean, depression is a big part of dealing with this disease, as it is for things like ALS and Parkinson's and Alzheimer's. So an increase in various biomarkers, including adipokines. Leptin went down, which you would expect with someone who's losing fat mass, but it went down proportionally more than you would expect. And adipoconnectin, I think, went up, and that's more of an anti-inflammatory thing. So they did baseline measurements and then repeated at 3 months and then 6 months. And then they showed, what was really interesting is that they showed fat mass went down significantly, and then there was a significant increase in related to overall body mass of fat-free mass. So of muscle, presumably, which
you
is,
can imagine in this patient population, that could be really important, even more so than the average person just looking to lose weight. We always want to preserve muscle, but like in a multiple sclerosis patient, especially.
absolutely. So I'm looking at the BMI. So they did lose weight, but they had the fat mass went from 44% to 40%, so of total body mass, where the fat-free mass increased from 56% to 60% of total body mass. So these are like effects that you'd expect with like an anabolic agent, like a steroid or something. So I think a lot of this had to do with just the dietitian stepping in, formulating, a modified ketogenic diet is higher in protein. So you have a very skilled dietetic team taking the wheel, ensuring that the patients are meeting all their nutritional needs. They were put on carnitine. The carnitine levels dropped even with a carnitine supplementation. And it made me think that, and I just had this conversation with Denise Potter, and that she puts everybody on carnitine. If you're burning tons of fat, you're using up carnitine and you could be carnitine deficient. So carnitine supplementation's pretty important. And but they had a variety, they had like 14 different tests, but the body composition was really surprising to me. And they had, so less than 20%, drop out, like 83% followed through. And they had significant improvement on a wide variety of tests, and we could go through all of them, but I think I'll just focus on the ones that are most relevant and kind of very interesting. They had the 6-minute walk test, which is kind of like a brisk, it's like a no nonsense test. You got like 100 feet hallway, and then the patient will just walk a brisk. They cannot run, they cannot jog, they walk at a fast pace, and you see how much distance they travel in that 6 minutes without walking and that was increased. Their quality of life in regards to mental health and physical health improved significantly. There's an expanded disability status scale scores, which is a variety of different things. And that's kind of like one of more like the hallmark, scales that are used for this. That improved significantly. You remember, Angela, we did the peg hole test for the NASA NEMO experiment. So they did the peg hole test for that.
Oh, cool.
And they were able to put the pegs. It looks at digital dexterity and reaction time, digital dexterity, and things like that, and just processing. So they did the 9 peg hole test faster. Serum leptin was lower. We talked about that. So what they found was that ketogenic diets are safe and tolerable, and they kind of knew that from phase 1. But this was a longer study. Phase 2 studies are traditionally a little bit longer. And body composition, fatigue, depression, quality of life, neurological disability, and various markers. They could have done a cytokine profile to look at the various cytokines and chemokines associated with that. Although many of the things that we have measured are not classically kind of associated with the disease. So that's but some of the things that they mentioned in the discussion is that the elevation and sustainment of ketones is important because that's linked to a reduction in inflammation. And inflammation, the autoimmune process that drives MS and the demyelination, is associated with a hyperactivation of the immune system. And there's data to indicate in multiple models that beta-hydroxybutyrate can reduce the activation and assembly of the NLRP3 inflammasome. There's that study, there's NRF2, there's NF-kappa B, and these things tend to be modulated by ketones functioning as a signaling molecule, independent of their effects of, metabolic physiology. And so there's that. And presumably, so some of the exclusion criteria for this, which I think is really important too, when you get patients on it. Let me bring up the paper here. So some of the exclusion criteria was, there was a couple people following the ketogenic diet already. So they were excluded from the diet as would be expected. 77 patients were screened. So they had 5 were due for loss due to follow-up, 3 were non-compliant with the ketogenic diet. So and then 2 withdrew from dietary side effects. So
Did they say what those were?
They did not say what they were, but I talked to some people, I think, associated with it, but it could have been due to just like GI discomfort.
Keto flu, stuff like that.
Yeah. Yeah. There could have been a little bit more information about how they tailored it to each. One person was diagnosed with Crohn's disease during the actual event or during, the study. But so they started with 77 patients that were screened, and they ended up, with 67 or 57 subjects were, ultimate KD compliance was 53. So 83% of the subjects met the compliance.
That's great. I mean, that's really good for a diet
this population.
Trial.
Yeah,
Totally.
that's
And
amazing, right?
The fact that I think it was something like patients showed nearly a 50% decline, right, in self-reported fatigue and depression, 50%. I mean, that's huge. When you think about probably some of the most key symptoms, we've not really talked about key symptoms, I guess, of MS. It includes such a wide variety of things, because this is where your body or your immune system is essentially attacking part of your body that is what covers your nerves, right? So think about what areas of the body that might affect. It can be so consuming, from fatigue to depression to tingling to numbness to headache. I mean, you guys, maybe Dom, explain some of those symptoms that you might see with an MS patient. And every single patient, I think, shows this. The reason why it's so difficult to diagnose is every patient shows symptoms very differently. So oftentimes it can kind of go undiagnosed for a very long time as well, based on symptoms.
Yeah. And this is more of a delicate population. So transitioning them to a ketogenic diet can be kind of tricky, right? When you have more of a fragile patient. So there's, as Allison Hall does, our collaborator on the CGM study in her clinic, is that she transitions patients over 4 to 6, because you can get a lot of side effects, but you can also exacerbate the disease process if you try to force someone onto a ketogenic diet from day one in macronutrients, and it looks like they took more of a transitional approach. The subjects were restricted to under 20 grams of carbs per day. One of the things that I've heard repeatedly is that electrolytes are super important. Calcium and magnesium are supplemented 500 to 600 milligrams a day of calcium. And a lot of blood work that I've seen with people show a decrease in magnesium and also calcium. And the calcium is, if dairy is removed and also vegetables and things like broccoli and stuff, or calcium intake can be low. So pretty much calcium is starting to be supplemented more often, and a reduction in dairy and stuff too. So they did a variety of different tests. They wore an actigraph too. So they didn't wear a Fitbit, but they did a waist-worn actigraph accelerometer to look at activity. Just to measure general activity.
Okay, cool. Did they see an improvement in that? Just because you could imagine that would be really encouraging to see that just in your daily life, someone who has these physical limitations are just able and intrinsically moving more, could be a quality of life benefit. That's
the things that trickle down effect from that too, right? And lead into other things.
Totally.
Yeah. So they were, their fatigue, they had a significant drop in fatigue. What was really interesting, they referenced a paper in fatigue and different strategies to reduce fatigue. And the ketogenic diet was effective as amphetamines for fatigue. So they had the same response in patients in regards to relieving depression and decreasing fatigue as modafinil. Often it's sold as Provigil. So that's the go drug everybody on the West Coast takes. The military uses modafinil as in place of amphetamines for a stimulant. So it was similar or or better reduction in fatigue than modafinil.
Wow.
And also amphetamine drugs. So methylphenidate and I think Adderall, which is like essentially amphetamine. So that's significant. So imagine the ketogenic diet and adding modafinil or something like that and relieving the patient. So there is that was pretty impressive to see those results reported. The Beck's depression inventory test, the BD1 test, significant effects on that. And so, yeah, I'm just coming to the point in the paper now as I'm looking through the text here. They did, and I think this should be replicated by other studies, that subjects provided a 6-month supply of the Bayer keto sticks, which is, I think, of an archaic ketone testing monitoring system, but they were required to take and photographed, and this is part of the compliance of the study too, to photograph their daily ketone test using a photographic evidence of each of the study teams. So apparently, I mean, I would think they would have someone ping them, send them a text in the morning. And I think there's a lot of things in here maybe that they didn't include to get that level of compliance, and probably a lot of training and education right up front to be able to do that, because people are going to read this paper and think, okay, I mean, this is going to work, and then they're going to try to implement it, but they don't understand all the time and effort and the team of people that is necessary to pull this off.
I will say we used a similar method through some of the pilot studies that I was involved with a variety of oncologists with cancer patients. We actually would have them take pictures of their ketone monitor with their ketone testing and their blood glucose, and also physically enter it into a system that when they were out of ketosis would actually message me on my phone. So I knew to check up with them right away. Right. So you don't have this like time lapse that goes by where the person kind of fell off the wagon and probably needs some encouragement. So you can kind of quickly get them back on. So I wonder if it's a similar sort of setup. And it was myself and a dietitian working very closely together. And it's a lot of work to be able to like train a patient to kind of go through those processes of, especially somebody who's dealing with something like MS or cancer, to be able to walk them through like, okay, taking pictures and then uploading here and then, and doing this each day. But I will say with some of these kinds of conditions, especially something like MS, where maybe some of these people it sounds like they're relapsing, really struggling and managing something like this terrible disease, they're probably coming from a place of like wanting to try something that might work at this point, and wanting to be an active part of the process. But I will say it is a lot of work. It does take a really diligent, committed team to be able to like get that granular with a process. But I will also say that the compliance that you get from that is, huge in terms of, maybe just having a study where they sort of self-report when they want kind of thing. It really does help a lot. Also in a patient seeing numbers and really being very conscious of what they're entering into the system, I think helps a ton in the process.
That's huge. I mean, that's all super important in what you just said. And well, I'll take a step back. So with MS, you get a constellation of symptoms, right? You get fatigue, you get reaction time, you get cognitive deficits too. I had friends that I know that had it, and it's just like they can't think. It's like brain fog, and then massively inhibited physical strength and reaction, the physical fatigue, but that strength really drops off too.
Well,
so
even heat intolerance, we were kind of talking about this. Temperature regulation is like a really difficult thing as well. And some of the management around MS is like stay out of the heat, stay out of extreme temperatures, because that can kind of send you into a potential flare.
Absolutely. So it's like, and I just mentioned a few things, but there's a constellation of symptoms, and then people can present in different ways depending upon where like the demyelination is happening in the central nervous system. But the first thing I did when I got this paper, I looked, ketogenic. I just immediately look, where are the ketone measurements? That was like the first 10 seconds of getting this paper. I looked at the graph to where that there are none, but then I was very biased getting into reading it. And then I realized, having talked to the epilepsy teams, and they're pretty confident that, and I've measured urine ketones quite a bit too. And I think it does. I think it's a good semi-quantitative way to confirm that you're in ketosis. But say, for example, if they had the Keto Mojo device, which has I'm not sponsored or, promoted, but they have an app, and then it does, glucose and ketones and the GKI for all that data to be uploaded like daily. And then people could report their symptoms or lack of symptoms in response to their ketones and be able to correlate that. I think that data would be like gold. I think maybe for phase 3, that would be like the perfect additional figure. And I think that would have made this paper like a super slam dunk. But I mean, it's an amazing study as is.
Yeah, I mean, I think all this brings up to me just thinking about the realistic, like practical challenges of having large clinical trials on ketogenic diet or dietary, studies in general. It's way more complicated than like a pharmaceutical trial. And you have issues with, compliance that you don't necessarily have with other trials. You have to have resources. It's expensive. You want to do it right. You want to be able to provide the meals, for the patient. That's really expensive to do. Even just, yes, of course, I totally agree that blood ketones are really gold standard in terms of just understanding a true reflection of the ketone level. But, I wonder what part of that decision was just, okay, well, it's expensive to run a trial and use all of this kind of more sophisticated techniques. And so, let's use urine ketones. And then that translates to people, you want this to be as translatable to people as well. So understanding where all of these different, practical techniques have their role. But, just thinking about also clinical implementation of this sort of therapy, again, it's more complicated. It really does require a team of professionals that can provide with constant kind of checking up and it affects everything, about their life just to make this huge change. For most people, it's going to be a really massive change in their lifestyle, and having the support necessary. It's just complicated. So, it requires that kind of oversight and especially in a vulnerable patient population.
Yeah. Yeah. And I'm just looking at, some of the limitations that they talk about too. I mean, randomization is going to be super important, but to do, this study was not blinded and is very difficult unless you have.
It's like, how could it be, right? Like, how do you truly
not with
blind?
whole food.
Yeah, exactly.
Yeah. Maybe with a drink, a shake supplement. Yep. Yeah. But some of the things that did surface on this was, the lipid profile showed a significant reduction in triglycerides. That's a big win. There was a significant increase in low density lipoprotein, cholesterol, and high density, but the ratio of the two did not change like all that much. And sorry.
Did they also measure things like CRP, sort of overall systemic inflammation, stuff along those lines?
No. So that's another area where I think, lost opportunity, we'll say, that they could have mentioned that too. I think there's a variety of different inflammatory markers that they could have measured, on that. But I think, just the effects on the fatigue and the depression and then the body composition changes are pretty remarkable. But I wanted to get to, and it looked like the percentage of fat, and I don't know, of the body like what the body composition demographics are for someone with MS, but the average body composition, or shall we say the average fat percentage was 40% for that. So, they started at 44% of fat mass, which is pretty high. And then they ended at 40%. And they were
at what? Sorry, Dom?
44%. Yeah, 44% fat mass.
Okay.
I think. And
how are they measuring body composition?
Yeah, I should know that.
Jackson Pollock or
Yeah. I think they did. Yeah, they did the bod pod.
Yeah, bod pod.
So, which, that's right. I have a star next to that. So, I think I did the bod pod a long time ago when it came up in like 2004 or something, when it was like this new thing, 2003. And yeah, it's a pretty reliable, method to assess body composition. DEXA would have been nice, but bod pod's pretty good estimate of body composition changes. So, but if they coupled this with some kind of exercise too, that would have been pretty remarkable. But they did a really good, variety of different tests to show the waist circumference significantly decreased too. I noticed that a lot of the magnitude of the beneficial effects were kind of experienced during the first 3 months. And then there were effects during, for example, like with the body mass index, about 80% of the effects were experienced in the first 3 months. And then it kind of tapered off. And a lot of the benefits, the majority of the benefits were experienced, but that's pretty normal with most studies, as was with our studies. But they continued compliance all the way throughout. And some of I want to get to the blood work, because I had a couple things on there, but the blood work was pretty, as mentioned, the carnitine levels dropped and vitamin D status went up. Hemoglobin A1C dropped a bit, but not too much. But the triglycerides dropped about 15 to 20%, which was pretty good. LDL.
Oh, sorry. Go ahead, Dom.
Oh yeah. LDL went up about 10%. So not that much. And then HDL just went up mildly.
How much did vitamin D go up? Because that's an interesting one, because I think there's actually been some research showing that adequate levels in folks with MS might result in fewer relapses. Yeah. So that is a really interesting piece of the puzzle that's already kind of being looked at. So I'm curious, like, what the difference was, if there was some report on that.
Yeah. So vitamin D nanograms per milliliter was 44. Which is like, almost like mid-range, it's pretty low. But then it was after 3 months, it was 11 points higher. So they're just putting in the delta. So 55 and then 12. So approaching about 60 nanograms per deciliter at the 6-month period. So that was statistically significant. I think they did. Yeah, they did include vitamin D to just ensure, it was like a small vitamin D. 400 to 600 units, which I don't know. I take like 5,000 or 10,000 units. So I was just thinking when I read that, that's not going to do anything. So I think it was probably just, they're just following proper nutrition. They have, oversight over their nutrition. And because when you look at the body composition alterations in response from baseline to 3 months to 6 months, these are something that you'd expect with some kind of performance enhancing drug, like the fat loss and then the lean body mass. They ended up, the lean body mass, percentage increased as they kind of related it to total body mass from 55 to 60. So that was really significant. And I think that's a super important biomarker, anthropometric biomarker to measure for this group. And I think as would, any kind of ALS, any kind of, Parkinson's, muscle correlates to, is directly correlated to, independence, but also brain function and also outcomes across the board. Mm-hmm. So,
I was just going to say their lean mass went up by how much? Because if their exercise wasn't in the mix, and you've got, some folks that are pretty sedentary, I wonder if that's more of like a pretty big shift in water weight that's influencing some artificial, you can see some changes in body composition that, sometimes some people are like, I gained 5 pounds of lean mass in a month, and it's not quite possible to do certain things without stressing the muscle and, a variety of factors. And so sometimes you can see some inflations of body composition that might not quite.
Yeah.
match
No,
up.
I'm glad you pointed that out, because that's exactly the case here. So they did like a clever representation of fat mass as 44 at baseline to 40% of total body mass. So they lost 4%. And then the fat-free mass went from 56% to 60% of total body mass. But I think there was a slight loss in muscle mass just because they lost a significant amount of their body mass, right? But they reported it as an increase in fat-free mass, but the increase in fat-free mass was proportional to total body mass.
It was the percentage.
Yeah, but that's like, and then they listed it. It was like, like 1 pound, or I think it's reported somewhere. It was pretty small. I mean, even if someone who's really into working out and then really starts training and loses to cut up, as in physique sports, and they're losing fat mass, you can't gain muscle and lose fat at the same time. I mean, that's extremely rare. I mean, it can happen under certain drug stacks and things like that, but that's pretty uncharacteristic.
You're a unicorn if that happens.
Yeah.
I would have loved to have known that in my professional career. Like, how do you do that simultaneously? And not to mention like multiple pounds of lean mass at the same time. Yeah. There's a lot of people who do that, but it's pretty, in my own experience, pretty difficult, and in training athletes. But yeah, Angela, what were you going to say?
Well, I was just thinking, but I wonder, in a unique situation where you have muscle, catabolism, and in the presence of ketogenic diet, and we know that, Dom, you were talking earlier about ketone signaling being involved in the anti-inflammatory side of things, but also knowing that ketones can signal through many pathways to slow or prevent catabolic processes of muscle breakdown. So, and then you also have, individuals who are regaining potentially some mobility. They're improving in these other aspects of even the quality of life, like feeling 50% better in terms of your fatigue or your reduction. You're going to be getting up and moving more. And that in and of itself is kind of like an addition of exercise. And so having that alongside something where ketone signaling might be helping support, preventing that muscle being broken down. And it makes me think of, I know there was a paper a few years ago looking at, a particular effect of, I think, acetoacetate in inhibiting MAP kinase signaling in skeletal muscle to prevent catabolism of the muscle in a model of, I think, muscular dystrophy or some similar kind of muscle-oriented catabolic condition. It's just cool to think about the multi-layers that are affected by a therapy such as the ketogenic diet. It's not a single effect. There's probably lots going on that could be supporting health.
True. Yeah, totally. A lot of different benefits. And like we were talking about earlier, like one small piece of movement influences like all of these other things that then they feel, that little bit of movement in the morning might brighten their mood, for the rest of the day. So they feel like they can eat a little bit healthier. They want to eat a little bit healthier. Right. It's like this ripple effect that you start with one thing. And so, you could totally be right of like, I think just the sort of muscle preserving properties that might be going on and what have you, and maintaining what they have. And then you add the layer of increased activity on top, you never know. But bottom line is, there's obviously no cure for this yet. This is something that affects 2.3 million people worldwide that don't feel like they have many answers, and many of which are relapsing even with medication. So, something like this kind of study is like the biggest and greatest step in the right direction for something. I just think in general, autoimmune disorders just don't have a lot of answers right now. And I think we can continue to expand the research on nutritional therapy specific to those kinds of disorders that we really don't understand at this point. And you see results like this, oh my gosh, makes me so excited. I
Yeah.
mean, somebody who suffered from long COVID, which is now by some considered an autoimmune issue triggered by a virus, much like what some theorize MS to be triggered by Epstein-Barr virus, to have some options around food that you can do sort of every day when you've got like all these crazy symptoms that just don't have any explanation. And for years on end, this is such a bright light for people who just probably haven't felt like they have a glimmer of hope for a while. So
Totally.
I think it's super exciting.
I totally agree. And I know we're going to put the link to the paper, underneath this, and that could be a resource for someone who wants, if you want to, talk to your physician about it, like print out this paper. It may be that they don't know that this kind of research is going on, and you could bring that to them, because a phase 2 trial that's showing these kind of benefits and in a disease that doesn't have many good options, I mean, that could really open a lot of doors. So it's a cool way to have that conversation with your healthcare team.
Yes.
Yeah, this is by far the best paper that I've seen that targets a nutritional approach to MS. And MS is also the prevalence of MS is geographically distributed too, from more northern regions, which are presumably less vitamin D.
Yeah.
So the vitamin D thing is something that's being studied now, but I think there's a big opportunity to study, and this gets back to the viral hypothesis, Epstein-Barr, maybe cytomegalovirus, other viruses, and also an infectious agent that could be causing this. It could be Lyme disease, right? It could be different mold or whatever. But I do think, as with Alzheimer's disease, that the etiology is largely unknown, for sporadic, right? Especially for sporadic. And I do think that more effort needs to be focused. And I think we're just not putting the top people who are studying these diseases into looking at that hypothesis that an infectious agent is being the trigger of this. I mean, there's a lot of clues out there that it could be. And I think for MS especially, that, and for ALS, definitely with ALS and Alzheimer's and Parkinson's and
it would make it so much easier if all this stuff wasn't so complicated, but like pulling in, the immunology side of it, like I don't know, it makes it hard to study these sorts of things.
It's the same with cancer, right? So you have, what was it, Albert Szent-Györgyi, the oncogenic paradox, where you have multiple different things that can lead to oncogenesis and multiple different factors. So it's a disruption of the microenvironment, but I mean, we're biased, but we think it is tied to metabolic health, and the health of the mitochondria will dictate the health of your central nervous system and will suppress oncogenic transformation, I think. So we are adamant about that.
We are pro mitochondria. You heard it here first. Taking a stand.
So this is about and this is about treatment, right? What do you do when you have the disease? But unfortunately, little or no funds are talking about prevention. But metabolic health is key to prevention. So
and that's why
especially can't be understated.
Go ahead.
Well, I was just going to say, and we're talking about Alzheimer's earlier, like that should be, I mean, we know the person, I don't have them off the top of my head, but the percentages of just a typical person, even if they don't have a known, gene that could increase their likelihood of getting Alzheimer's. I mean, once you get to a certain age, that risk can become quite high. And so it should thinking about these early prevention, especially lifestyle-based changes that people could be making in their 30s and 40s that could dramatically impact, whether or not they end up developing Alzheimer's before they pass. It just makes total sense as a huge, we should be putting a lot of effort into this.
Yeah. And that's why we're doing what we're doing, trying to bring the right people in the room, right, through Metabolic Health Summit. And then also something like this podcast, having a discussion around these topics, I think is so important. And also to those who are listening or watching, engaging with some of this information too, right? Asking questions, posting, sharing, writing a review for this podcast as well, just helps us get into more eyes and ears so we can kind of spread the great research that's going on right now, and hopefully accelerate the process really of and bring the right people together that might hear this, or even going to your doctor, as Angela mentioned, and bringing this paper to your physician's attention. Even that step can literally change the trajectory of a physician's career. I've seen it happen where they learn about this research and they're like, oh my goodness, why is this not why am I not implementing this into my practice? And that's the start of something really special. And honestly, we've got many physicians that show up at the conference that start in that way, where a patient came to them. So I think there's so many important people in this whole process, from the medical world to the academics to the lay public who are tuning in, and everybody together is really going to make a difference in moving this field forward. So with that, I'll say thank you guys for the great discussion today. And thank you all for listening to this. You can find the paper links, as Angela mentioned, in the description of this episode. And we will continue to be putting out research on metabolic health and metabolic-based therapies here very soon. But thank you guys for hopping on today. And until next time, we'll see you all very soon. Make sure you share this podcast, like, review, comment, all the things. And until then, we'll see you next time. Bye, guys.
Bye.
Thank you.
Thank
Bye.
you. See ya.